The sequencing of tumors has revealed thousands of genetic alterations present within them. The challenge now is to determine which of those changes are actually important for the development of cancer and which are not. You've probably heard of trying to find a needle in a haystack, but I bet you haven't heard of finding one specific needle in a stack of needles. This is what the researchers at the Cancer Research Center of Salamanca set out to do. In their laboratory, they focused on a specific mutation of a gene, in hope that this would be one of the mutated genes which holds significance in the search to better understand cancer, its origins, and, ultimately, how to treat it. Could this needle be the one they are looking for? This gene, denominated RAS2, is very structurally similar to RAS proteins, which is particularly interesting because these molecules are frequently altered in many human tumors. Normally, these proteins shift from an inactive to an active state. The activation step is mediated by extracellular signals, leading to the generation of intracellular messages that tell the cells that it's time to start proliferating. However, when mutated, these proteins are continuously in the on state. And as a consequence, the cells start to proliferate without any control and eventually form tumors. In order to find out more about these mutated proteins and their correlation to cancer, the Cancer Research Center of Salamanca arranged a study on genetically modified mice in which they could induce, at their discretion, the expression of the mutated version of RAS2. Through this process, the researchers were able to mimic perfectly what happens when these mutations appear in humans. After the induction of this mutation in mice, many different types of tumors appeared. This mutation induced profound changes in the signaling pathways of cancer cells. In addition, it was found that those changes varied depending on the type of cell that harbored the mutated version of the protein. This data unequivocally indicated that the RAS2 mutations found in cancer were tumor drivers. The further analysis of these mice allowed the identification of a number of therapeutic targets in RAS2-driven tumors. As a result, the investigators could identify specific anti-tumor drugs and combinations of currently available drugs that could be used to specifically eliminate the tumors generated by the mutant version of RAS2. These results suggest that the administration of such therapies could be useful in the case of patients that bear tumor-promoting mutations in the RAS2 gene in their cancer cells. These therapies could be also useful for other diseases in which activating mutations of RAS2 have been recently found. Although more work remains to be done to elucidate all the factors that contribute to the development of all human tumors, this work has pinpointed a critical molecule that promotes the rapid development of specific tumor types in our body. It has also unveiled key vulnerabilities of those tumors that could be used in the future to treat patients with specific anti-cancer drugs.